Hello everyone, I'm Ashani Sarkar and I'm going to present a topic from medical microbiology, which is conventional lab diagnostic methods. So let's get started. Introduction. Medical microbiology is a branch of medicine that deals with the prevention, diagnosis and treatment of infectious diseases. In addition, this branch of science explores various clinical applications of microbes to improve health. There are four types of microorganisms that cause infectious diseases. They are bacteria, fungi, parasites, viruses, and a class of infectious proteins called prions. Medical microbiologists study the properties, pathways, and mechanisms of transmission of infections and pathogens. Principles of lab diagnosis. The diagnosis of microbial infection begins with the evaluation of clinical and epidemiological symptoms, which leads to the formation of a diagnostic hypothesis. Physicians dealing with an infectious agent need to know when and how to collect specimens and what laboratory tests to order. Morphologic identification of agent, culture isolation and identification, detection of antigen from the agent, DNA or DNA-RNA hybridization. Now the first one here is basic microbiological techniques. So hanging drop method. Preparation of hanging drop is one of the easiest ways to observe motility in clinical microbiology laboratory. This is done by placing a ring filled with reagent and the bacterial suspension on a glass slide, placing it in well glass and observing the inoculated saline under a microscope. The advantage of this method is that live bacteria can be seen through a cover glass. Studying living organisms is useful for monitoring cellular activity. Bacterial motility can also be determined by the Craig tube method, bacterial colonization on a non-inhibitory method, and microscope in dark method. Isolation of pure cultures. So to achieve discrete isolation of colonies and pure cultures, the number of organisms in the initial inoculum must be reduced. Further reduction in population size ensures that individual cells are clinically sufficiently distant from the surface of the agar medium after plating. The streak plate method is a rapid qualitative separation method in which loop full of culture are spread over the surface of an agar plate. So the spread plate method is used based on spreading bacterial colonies on the surface of solid agar medium while rotating a potato and turntable using a sterilized L-shaped bent glass rod. The pour plate method is a fast quantitative separation method where the method requires serial dilution of the mixture culture with pipette. This method involves pouring sample of the molten agar into a petri dish, allowing it to solidify and incubate. Purity of isolated colonies can be only verified by carefully transferring well isolated colonies of media by streaking. Now let's talk about bacterial staining techniques. First one is gram staining. So it is originally developed by histologist Christian Graham in 1884. The gram stain is a method of staining bacterial tissue. So gram positive bacteria stain purple and gram negative bacteria stain pink. They are colored differently due to significant difference in the complex light macrostructure of the cell wall. About 60 to 90% of the cell wall of the gram positive bacteria composed of peptidoglycan and teichoic acid, and only 10 to 20% of the cell wall of gram positive and free negative bacteria is peptidoglycan. The gram negative cell wall is also methyl violet, surrounded by an outer membrane composed of phospholipids, ethanol, lipoproteins and other proteins. Now let's talk about acid fast staining. So acid fast staining method is a modified version of the method of Ehrlich. Ehrlich first proposed a method for detection of genesian and concentrated nitric acid. Conventional aniline dye solutions are not suitable for staining because of their low permeability to Mycobacterium tuberculosis substances. The acid Acid-tolerant Zeal-Neelsen stain has been found to be most useful for staining acid-tolerant bacilli of the genus Mycobacterium, particularly Microbacterium tuberculosis and Mycobacterium leprae, as well as Nocardia bacilli. These acid-fast bacilli have high concentration of lipomycolic acid. Cell wall painted, it looks pink or blue on background. Now let's head over to biochemical techniques. So first, catalase test. Chemically, catalase is a type of hemoglobin that is structurally similar to hemoglobin, except that 4 iron atoms in the molecule are in the oxidized rather than the reduced state. This enzyme converts hydrogen peroxide into water and oxygen. Catalase production can be detected by adding hydrogen peroxide to the bacterial colonies, and presence of catalase indicates the presence of free air bubbles. In the absence of catalase, bubbles are not visible. The catalase test is mainly used to determine the members of the family Micrococciae from the members of the family Streptococcus. A mycobacterial catalase test is also performed to distinguish Mycobacterium tuberculosis from atypical Microbacterium. Coagulase test. So, coagulase produced by a small number of staphylococci. It is a major characteristic of pathogenic staphylococci. The enzymes cause the blood to clot, allowing the body to effectively disconnect the infection from the host defense mechanisms. Coagulase is a protein with prothrombin-like activity that converts fibrinogen to fibrin, which forms visible blood clots. Under laboratory conditions, the coagulase test is used to determine distinguish Staphylococcus aureus from the other types of coagulase negative staphylococci. Now next we will move on to antimicrobial sensitivity test, which is Kirby-Bohr method. So with the emergence of many antibiotic resistant bacterial strains, antimicrobial susceptibility testing is performed to determine which antimicrobial agent should be used against a particular bacterial strain. Available chemotherapeutic agents vary in their spectrum of antimicrobial activity. Some have their limited spectrum, while others have a good spectrum antibacterial activity. Bacterial strains resulted from clinical specimens should be tested for susceptibility to antimicrobial agents. This is because it can tell the doctor which antibacterial therapy to start with. Now let's move on to the immunological tests. So few immunological tests, and among that I have choose two, which is first one is ABO blood grouping. So ABO system consists of 4 blood groups determined by the presence or absence of 2 different antigens A and B on the surface of the red blood cells. Group A red blood cells have A antigen, group B cells have B antigen, and group AB cells have both A and B antigen. Group O has neither A or nor B antigen. The 4 groups are also distinguished by the presence or absence of 2 different allo antibodies in serum. Serum contains allo antibodies present for the antigens not present on the red blood cells, blood grouping antigen inherited according to Mendel. So now the next immunological test is viral test. So the viral test detects antibody against Salmonella paratyphi A and paratyphi HNO suspension in serum. Because S. paratyphi C infection has not been found in India, and serum antibodies against this type have not been tested. Serum of this tested it is a test sealed diluted in a test tube containing suspension of Salmonella antigen. These 4 solutions are prepared at S. typhi antigen known as typhi H antigen discontinued. Test is then incubated 37 degrees Celsius water was checked for agglutination by checking the sediment at the bottom of the tube.